unless and until you analyze them, and unless and until you find out what are the ingredients present there, and what is the concentration. Say for example, another very important issue is that water quality. This measuring the arsenic concentration, that also comes under this analytical instrumentation. Okay. And for industrial application, there are plenty of applications in the industry for gas analysis in the refineries or in the power plants for water quality analysis of boiler. Uh, and in the medical industries, say for example, and another very trivial example I'm giving you. Say we take this paracetamol when we have fever, right? So paracetamol you get 500 mg or 650 mg or 1000 mg. So, but the tablet, the size remains same. Okay, so what is there? See, in that tablet, the concentration is more. So if you want to measure what is the exact value of what amount of paracetamol is present in a tablet, you need an analytical instrument for that. Find out whether it's 500 mg or 650 mg. And there are some application related to our related for our, and that's used in this pathological laboratories. For example, if you want to measure the sodium or the potassium or our blood sugar, all these instruments, they come under this analytical instrumentation. So you see the application area is huge, and these analytical instruments are not only used by the industry people in the refinery or in the power plant or in the steel plant, but they're also extensively used by the different laboratories. For example, and all the chemistry labs, they use this chemistry labs or the physics labs. They use this for different type of applications. Chemistry people, they use it quite extensively. The biology people, the scientists who are working in different labs. So there are, the area is huge, but definitely we'll concentrate more on the industrial point of view, and at the same time we'll discuss the other types of instruments. Okay. Now to start with, I think, let me share the screen. I have written I do not have any PowerPoint presentation, but I'll definitely show some videos. Yeah. Now, can you see the screen?
Yes, sir.
Okay. So you see, I've written just like in a blackboard, the way we write. I have written in a sheet of paper and taking the photograph. So this is in a nutshell what I've mentioned. It deals with the analysis of mixtures. It can be gas. It's mostly related to gas or liquid or solid. Also is there. Sometimes we need to find out the composition of solids, particularly in say steel plants or in other say where the metals are extracted. It is used for not for quality assessment of different products. So unless and until you analyze the compounds or the product that you are manufacturing, you cannot ensure the quality. So you need this type of instruments to find out what are the different compounds or the different elements present in that uh, particular product. So, and that is also true for quantitative assessment. That's what just I exactly mentioned about this paracetamol tablet. So there are a number of applications, measurement of blood parameters, blood sugar, urea. It's just a repetition what I've told. Quality assessment of petroleum products. This refineries, they use this analytical instruments quite extensively for gas composition or for say the petroleum is basically a mixture, right? So when you pass it through distillation column, you need to analyze and then take the proper measures. Then another is the pollution measurement and control on the air quality and other type of pollutions. Okay. And then water quality. Water quality is, for example, we have nowadays these RO water filters in our house, and you'll see that the person who maintains that, they come with a probe, and it measures the, it's called the TSD, sorry, TDS measurement. So that probe is used for total dissolved solids in the water. So that gives us an idea about how pure the water is. So for drinking water, it is used, and this is also true for the water going to the boiler, because if we, I mean, allow water with many elements and compounds inside it, uh, to the boiler, so there will be corrosion, there will be coating inside it, and we have to replace the boiler tubes quickly. So we have to ensure that. And wastewater streams also, there we have to measure different compounds, whether they're, polluting the environment. Okay. And oxygen concentration in combustion processes. This is also used in the power plants, because there the air fuel ratio is to be maintained. Now, in order to maintain in the combustion process, the air fuel ratio is to be maintained at an optimum value, so that we get the maximum efficiency out of combustion. So you need to measure the oxygen concentration in the air, which is going as the coal or other fuel. Okay. So these are some of the application areas of analytical instrumentation. And this analytical instrumentation, there are three main categories, although there are many other techniques which also come under this subject. But primarily, we'll discuss these three categories, and you'll see perhaps more than 80% of the instruments, they come under either of these three category instruments. So the first category instrument is the separation instrument. Separation instrument means that they are based on the principle, principle that will separate. If a mixture is composed of three compounds, then it separates by some means. For example, you have studied this gas chromatograph in your school. You have studied what is this gas chromatograph or this liquid chromatograph. There in a column, the different components are separated, and after separation we can measure it with some detector, or another very useful instrument is known as the mass spectrometer. Mass spectrometer is also based on the principle of separation, and there the different compounds or the different components, they are separated based on their mass by charge ratio. So they are charged with the help of electrons, and then that particular component, they are separated. They come, they pass through a column, or some tube, and they appear at the detector at different times. So this is one class of separation instruments, and these and mass spectrometers are used extensively in the research labs, and mainly in the refineries. And we have recently procured one GCMS system in our lab. And when you come to the college, we'll be able to show you how this separation takes place, or we can give you some demo also here. And then the second class of instrument is the electrochemical instruments. Now, you see, this analytical instrument is such a subject that it requires a lot of knowledge, all of basic knowledge about physics and chemistry, and since it's mostly used by the chemists, there's a lot of chemistry involved in chemical interpretation. But as an instrumentation engineer, we really do not need to know about a little bit of chemistry, the basics, but not extensively. And interpretation of this output from this instrument, that is usually done by the chemists or the domain scientists, biologists, or physicists. Now, these electrochemical instruments, they are also very, extensively used for different purposes. One is the pH measurement. pH, do you have any idea how pH is measured? What is the basic principle of measurement of pH? Anybody?
So hydrogen ion concentration.
Right. And there is one electrode used for that sensing. What is that electrode? What is the composition where of that electrode? What is there? No idea. It's basically a glass electrode, and this glass electrode is being used for the last 40-50 years for measurement of pH. And if you glass it with a certain composition in a liquid, then there is a hydrogen ion. I mean, the concentration of hydrogen ion is different at the two sides, and because of that, there is a voltage generated, and that is governed by the which equation that also we have studied, or electrochemistry.
Nernst equation.
Yeah, Nernst equation. So Nernst equation is the governs how much voltage is generated. And this electrochemical instrument, this is one example of the pH, and there are other examples also. pH is basically it's a potentiometric type of measurement. That means the open circuit voltage is measured. Now this pH measurement was in earlier days it was really very difficult because this glass electrode, the impedance is really very large. It's of the order of giga ohms or several mega ohms, and it's not so easy to measure the voltage, the millivolt that is generated in a pH electrode. So but nowadays we have this instrumentation amplifiers or other type of high input impedance amplifiers. So with the help of that, it can be measured pH. So pH meters are available and they're not at all very costly. Now so this is basic. This is based on the principle of potentiometry, but there are some other techniques. For example, amperometry. Amperometry means that we apply some voltage and current is allowed to pass through the solution. Okay, and that current will give you an idea about how much what is the concentration of that particular analyte. And this amperometric measurement is used in our glucometers. You must have seen most of you. It's for measurement of blood sugar. So when the drop of blood is given to that meter, then there's a current through it, and that current will give you what is the amount of blood sugar present in that particular sample. There are many such electrochemical instruments, electrochemical sensors, which are available. Which these are also used for sensing gas, not only gas. And you will be, it's really. There's another example which is really not very easy to understand. That these electrochemical instruments, it's based on Nernst equation on the basic theory of Daniel cell is there. You understand it well. And whenever we talk about electrochemical instruments, we usually think that it's in a solution. So we dip an electrode and we generate some voltage or some current is flown, and then that is measured, and that will give you the idea of the concentration of that particular analyte. But in one application, and which is used quite extensively, it is for the solids. And it's for oxygen measurement. There is no liquid solution. The solution that is used is, and that's basically zirconia oxide. And zirconia oxygen analyzers are very popular, although they're quite expensive. And but they're quite extensively used for measurement of oxygen, mainly in the power plants or other where the combustion processes are there. So there's a large number of applications for electrochemical instruments, and in many cases, these electrochemical sensors are quite inexpensive, but they are very robust and they give very accurate reading. So they're very popular. And the third category of instruments, some of you have already got the exposure. That is the spectroscopic instrument. It's based on the principle of spectroscopy, optical spectroscopy, optical or other type of spectroscopy is also there, and we'll discuss about them. And spectroscopic instruments, although they were mostly used in the research labs, but now they are in the field, and you'll see the spectroscopic instruments are used in the plants for measurement of gas concentration, and many other applications in industry as well as in the lab. So optical spectroscopy, it includes this UV visible, that's in the range where the signals are operated. Then we have the near infrared spectroscopy, then IR spectroscopy, then Raman spectroscopy that is also coming in a big way, because nowadays the components are becoming less and less expensive, and there are many such applications. NMR is another very useful useful spectroscopy, particularly for the chemists for finding out the chemical structure. NMR stands for uh, anybody, any guess? N for
So is
nuclear.
it nuclear magnetic resonance?
Nuclear magnetic resonance, right? Absolutely right. So NMR is nuclear magnetic resonance, and this is although this involves a huge electromagnet, and that's why it's quite expensive. It's used only in the laboratories, and very few laboratories can afford to have an NMR. And but this is very useful. It is not. It was discovered only in the 1940s, and but after that it has become very popular among the chemists, and in particularly in analytical instrumentation. Whenever you get a problem, or particularly the chemists or the biologists, they face a problem to find out a solution, or the find out the what are the composition. Only one method may not be sufficient, and one method, one has to, I mean, do a lot of iterations here. There are hit and trial methods that change the different settings, and they use the data from different instruments, and then after compiling the data from different instruments, then only they can interpret that this is the composition. Okay, but NMR is really a very powerful instrument, and another offshoot of NMR is you all know, that's the magnetic resonance imaging. That is for finding out the cancerous cells in our human body. So you see, these are the three categories of instruments: the separation, electrochemical, and spectroscopic instruments. And apart from these three, there are many other instruments which are also used for analysis purposes. And there, say humidity, then turbidity, how turbid a solution is, then conductivity, then viscosity. For example, say if you want to measure the wastewater quality, then you need to measure conductivity, you need to measure pH, you need to measure the turbidity, and maybe a few other parameters. Okay, so all if you, I mean, publish all the parameters, then only the pollution control board will accept it. So this is in a nutshell about the introduction of analytical instrumentation. And regarding reference books, the book of Spook is excellent. It is mainly the theoretical and basics are there. Almost all the topics are covered in Spook. And Kanpur is also another very good book. It is written by one Indian author. And LibTAC, there is one separate, section is there for analytical instrumentation. So I'll take the material from all these three books. Okay. So now today we'll start with the basics of gas chromatography. And gas chromatography, it's basically a technique. But I think let me show one video. I think it will be easier for. This is available in the YouTube, but it will be easier for you to understand what is just, or you must have studied it. And, yeah, can you see it?
Yes, sir, it's visible.
It's visible. Okay, very good. So now I'll start it. Yeah, actually, at this point, I'm interrupting. The stationary phase, there are actually these two terms, and they are used in gas chromatography. One is called the stationary phase, and the other one is called the mobile phase. The idea, it has come from the fact that whenever in a particular column we inject the composition or the mixture, then the different components, they are absorbed, and they take some components take longer time to pass through. Some components take a smaller time to pass through. So, and it is mainly because of the selective absorption of a material which is coated on the column. So that is known as the stationary phase, because the input sample is stationary in that particular material. And the mobile phase is the gas which is pushing the mixture through the over the stationary phase. So that's why this stationary phase and this mobile phase, these two terms are there. Okay. So is it clear to you? Okay, let me. Yeah. So I think you could understand it very easily, although it's a very simplified presentation. In fact, it is shown here that there are two types of columns. One is the packed column, and the other one is the capillary column. And it's easier for us to understand what is the packed column. Packed column, it's the stationary phase materials, and they are packed inside the column. And the mobile phase material, it pushes the sample through the column, and at a particular pressure. So you need to give more pressure so that the sample passes over this stationary phase. But these packed columns are no longer used nowadays because of several reasons. One major reason is that in a packed column you need more uh, pressure, whereas in the case of and separation is also not very good in packed columns. Compared to that, the capillary columns, they're very, the diameter is very small, and that is that, and but it's very long. And since there is a small hole inside it, it needs a much less pressure. Okay. So nowadays, all the gas chromatographs, they use these capillary columns, and there are different types of capillary columns that perhaps I'll show you today. Can you see read from this screen now? Here are some definitions.
Sir, can you please zoom a little?
Uh, I think now you can see the distribute and the partition coefficient. Anyway, I'll just explain. It's really not very difficult to understand, but you have to remember these definitions, and perhaps I have to use some other pen for writing. Anyway, on the next class, I'll use some other pen. The first definition is known as the partition coefficient. This is the ratio of the concentration of the species in stationary phase and the mobile phase. So K is the partition coefficient, and higher the K, better is the separation. That you can understand, because if the amount is more in stationary phase, that means it is better absorbing material. Then the retention time. Retention time is most important. Retention time is nothing but the time that it takes to travel through the column. Or there is a this type of chromatography is known as elution chromatography, and the term is used as is elute. So it is the time required to elute a component from the stationary phase, and this retention time is the only parameter from which we can identify which type of compound it is. From the video, you have seen that you can identify from the retention time. And for a particular column and for a particular operating condition, this retention time versus the compounds, there's a big library available. So it's really not at all difficult to find out which type of compound is present in the there. So the standards are there, and they're regularly phase, or the mobile gas, or the helium gas, it pushes, it takes out the compound from the stationary phase. Okay, so this way, so gradually the sample passes through the column, and because of the different partition coefficient for different components, the compounds are separated. So this is known as elution. Elution means coming out. It takes out from the stationary phase. So this is the time required to elute a component from the stationary phase when the entire when it passes through this the entire column. Okay, so it's assumed that the entire column is composed of the stationary phase. So the total time is known as the retention time. And you see the relationship is V is equal to F into T, and where F is the flow rate and time T is the retention time. And for it, there are some other retention volume is the volume of the gas, mobile phase gas, required to elute a component from the stationary phase. Okay. So, and the dead volume is the time of an average molecule to pass through the stationary phase without any retardation, is known as the dead volume. So, if a particular a compound or a compound is not actually absorbed in the stationary phase, for example, the mobile phase, then it takes some time to travel through the color. So that is known as the TM, or and TM multiplied by the flow rate is the dead volume. So these are some of the definitions related to gas chromatography. And then so a chromatograph consists of the following parts or components: one, a carrier gas supply with a pressure regulator and pressure indicator. So the carrier gas, it comes with some pressure, and the pressure is quite huge for packed column, and it's much less for the capillary column. Then you have the sample injection system. Here, this is the injection system. Mostly, in most of the cases, we have the septa. Septa, it's a small, disc, a very small disc of say diameter. How much it will be? It will be say maximum 5 millimeter diameter or so, and it's made of rubber. And we push the injection syringe through this spectra. Some of you must have seen. You must have seen it while taking the some injection from a particular from a container. We insert the injection needle through a rubber. So that is known as the spectra. So through the spectra, through the septa, we push the sample. And here there are many issues. One is that the amount of sample should be very small. We should give very small amount of sample in the injection port. There are several reasons of that. Only few microliters are given. And this injection port is at a higher temperature usually than the column. Not only that, it is to be ensured that since it's a gas chromatograph, the injection port at the temperature of the injection port should be such that all the components of the mixture should be in the gaseous phase, or the temperature of the injection port should be at least higher by 20 degree or 50 degrees centigrade than the highest boiling point of the composition of the mixture of the components of the mixture. So injection port is one component of gas chromatograph. So you see, you have the pressure regulator, pressure gauge, then a rotameter or a flow meter for measuring the flow rate, and then this is through this part the mobile phase is coming, and through this part the sample is coming, and this is the column. Column is usually in the form of a coil inside the gas chromatograph. So when you come to the college, I'll show you one column, and then it is kept in an oven. And one point it was mentioned in the video that the temperature should be maintained at a particular value, but this is always not the case. In most of the cases, we give a ramp, and that is known as the development of a particular method for separation. Say for a particular mixture, you need to ramp at a particular rate and vary the temperature from say 150 to 250 and so on. So this type of methods are developed by the chemists or the experienced person who are handling the system. So they know that for a particular mixture or for what type of temperature gradient should be there. Usually it is not kept at as constant, because if you keep it as constant, then the separation will not be very good. Our main idea is that since the gas chromatograph is used for separating the components, we must ensure that at the exit all the components are separated. If they're mixed, even then there is no point in using the gas chromatograph. So we have to maintain the temperature and the temperature and the gradient accordingly. And after that, we have the detector. See, this is the detector. This is the detector. This is just a clock, but in most of the cases, the detector is outside this. This is the one type of detector that we have seen is the flame ionization detector. So flame ionization detector, it ionizes the compounds in a flame, and then it detects the concentration. And apart from flame ionization detector, there is another type of detector that also I'll discuss, that is known as the thermal conductivity detector. So that detects based on the principle of based on the thermal conductivity of the particular gas. So there are different advantages and disadvantages, but FID is very popular. Although the flame ionization detector, it is risky in the sense that it needs hydrogen and oxygen, and there is some the flame generation is there. And after that, it's a strip recorder or a computer, or where you get the entire the output. Okay, and this output is known as the chromatogram. The chromatogram. What will be the x-axis and what will be the y-axis? It's basically a graph. Okay, so there will be x-axis and y-axis. Any guess?
Distance and concentration or something like
Uh, Actually, on the
So time and
x-axis,
concentration.
time and concentration, yeah, and that's correct. And it's not exactly the concentration, it's the actual number of molecules which are coming out on the y-axis. And on the x-axis, you are correct, it's time. And this x-axis, the retention time will be different for different components, and we can identify the components based on the retention time. And the area under the curve will give you the number of molecules which are coming out from that column. Okay, so that is known as chromatogram. Okay. Just I'll give you a very small introduction to this plate theory of chromatography, and in next class I'll discuss some of the problems or how to calculate the number of plates. Now the plate theory, the theory of chromatography is really very interesting, and it is defined in this manner, or there are some theoretical computation. That means if you want to find out whether a particular column, how much efficient the column is for separation. So in that case, there are some computations. So we'll do that problems in the next class. And this is based on the theory of, it's known as the plate theory, right? And it is, I think you can see it. A chromatographic column can be considered as composed of a series of discrete but contiguous narrow horizontal layers called theoretical plates. There are no actual plates. It is those who have got any idea about the distillation column. I think I've studied in your school days. The distillation column, where the different components are separated in the refineries, and it is almost similar to that, but here there are no distinct plates. All these are it's a continuous, but theoretically we can assume that there are a number of plates, and which are contiguous and narrow, and they're horizontal. At each plate, equilibrium of the solute between the mobile and stationary phase is assumed. Movement of the solute and solvent is then viewed as a series of stepwise transfer from one plate to the next. And the efficiency of a chromatographic column is measured in terms of the number of theoretical plates n, and the second term is the height equivalent of the theoretical plate, that is H. So anyway, so L is equal to the total length of the column can be expressed as L is equal to N into H. So from the chromatogram, we can find out what are the number of plates in a particular column, and this is one of the important specification of a column. If you want to buy a gas chromatograph or a column, then you have to specify that what is the number of plates per millimeter or say per centimeter. Okay, so I think. I'm stopping this sharing and so any questions on what I've discussed till now on gas chromatography? There are other chromatographic techniques. Gas chromatography is for separation of gaseous compounds. So similarly, we have the liquid chromatographs for separation of liquids. Okay. And the basic principle is also same. There is a column, and inside the column, the separation takes place.
Sir, how efficient is the gas chromatography?
How?
Efficient? Like
Uh,
we're dealing with gases, so I think it won't be that good enough.
No, actually, this type of these gas chromatographs are used in many applications. For example, say in a mixture of and say natural products. Say, for example, I have used this gas chromatographs for tea volatiles. Okay, and tea volatiles, there are a large number of compounds. Okay, and the problem is that is how they are separated. There are many issues. One is that you should not give. I mean, the sample size should be very small. If you give too much of sample, then it would be very difficult to separate them. And there is another issue. If you give too much of sample, then there is a concept called band broadening. See, when a particular compound is eluting from the column, or when it's coming out from the column, then the number of molecules should be as low as possible. Because if you have large number of molecules, they will come out with a longer time, and there is a possibility that there will be overlapping between the two components. So in that case, the separation will not be good. Now, in order to have a good separation, the method is to be developed for liquid chromatograph or for gas chromatograph. You have to. It is called method development. What is meant by method development? Method development is nothing but you have to find out, say, or for a particular mixture, and for a particular column, what would be the temperature gradient or the temperature change. In many cases, we have seen that the temperature changes, say, from first 5 minutes, or say first 2 minutes, say from 150 to 200 degrees centigrade. Then the temperature is kept stable for 2 minutes, and then the temperature rises from 200 to say 300 degree in another 2 minutes. So this type of variation is done by the experienced operators, and they know that if we change the temperature in this way, then the separation will be good. In any case, it's possible to separate the different components in this gas chromatograph. And we have another very powerful instrument that is known as the mass spectrometer, that is also used for the separation. But the difference is that in that case, the separation is based on the principle of mass. Okay, that the molecular mass that actually decides the separation. But here it is not exactly the mass, but it depends upon the this how good it is getting absorbed in the stationary phase, and how long it's taking to come out from the stationary phase. So this gas chromatography, this gas chromatography is in use for quite some time, and because of that, the methods are already available for most of the mixtures, and they are widely used. And there is no issue of efficiency. It's definitely a very good, very efficient machine in sewing. Okay, Devanshu.
Yes, sir. Yes, sir.
Okay. Any other issue? The basic principle is really very simple, but there are many a large number of applications. Okay, I think you go through the books. The book of Skoog is there. I think if you can manage an ebook from some sources, that would be wonderful. And there are some YouTube videos also. That the one I assume that is good enough for understanding the basics of GC. And definitely will show you when you join the university. I'll give you the demo here in our lab. Okay, so if there are no other questions, then well, thank you very much for attending this class, and then the next class will be
One query, sir.
sorry,
স্যার ওই বইয়ের নামগুলো যদি আরেকবার বলে দেন তাহলে ভালো হয়।
বইয়ের নাম একটা হচ্ছে স্কুগ, SKOOG, আরো দুজন অথার আছে, হ্যাঁ, অন অ্যানালিটিক্যাল ইনস্ট্রুমেন্টস বা এরকম একটা নাম, সরি ইনস্ট্রুমেন্টাল অ্যানালিসিস বা এরকম একটা নাম হবে, হ্যাঁ, স্কুগ। তারা আমার কাছে বইটা আছে, আমি দেখাচ্ছি এক মিনিট। এটা তো অবশ্য অনেক of ইনস্ট্রুমেন্টাল অ্যানালিসিস, হ্যাঁ, আর এটা তখন যিনি লিখেছিলেন শুধু স্কুগই লিখেছিলেন, I think, but there are I mean other authors. And Skoog is a very famous scientist. He's from Stanford, so it's a very excellent book. I mean, you will really, if you go through the book, most of I mean, you cannot go through the book full, because there are so many other things, so many I mean topics which are mostly for the chemists, not for us. But anyway, it has dealt the instrument part well. And there's one, the Indian book is also pretty good, that Kanpur, KHANTPUR, and that book is also pretty good. So you please try to find out if you can get a copy, and it's excellent. Otherwise, you can buy that book also. This Kanpur, or this will be very useful. And LibTech, that portion is also needed. There are many industrial applications which are discussed in LibTech. Okay, so our next class will be once again on next Friday at this time. Anyone has got any issue regarding this Chhat Puja? Because the subject is huge, although I don't know in this situation how much I can cover, but we have to try. And I'm not very sure how long this university will remain in this situation. So,
Sir, is there any chance to open on December?
I don't think so. I don't think so. You mentally be prepared that this semester will be totally in online mode. So try to, and you have got many important papers in this semester. So
Yes,
try
sir.
to learn on your own. I understand the difficulties that you are going through, but anyway, we have really we can't do anything. First, we have to be safe enough, and then our studies and all these things. So collect all the books, and we can discuss over this web meeting, and we can discuss at any other time also. There's one advantage of being accustomed with this type of environment, and we can discuss at late night or early morning. There's absolutely no issue. So if you do not have any issue, Shayan, you please discuss with your friends. If there is no other issue, then we'll once again meet at least for the next class next Friday at 3.
Okay, sir. Okay, sir.
And you send the link. And if there is an issue, then we can take it on Saturday also. If there's any, because the Chhat Puja is there. Some of
Sir,
you might...
I will inform you. Sir, I will inform you on this regard, sir.
Okay, Shayan. Okay. Thank you then. I'm leaving.